We should get started. It's great to have Paul Garofolo here today for another segment of our podcast, and we're excited to talk to Paul because he has an interesting perspective on phage therapy, and we like to get those interesting perspectives and have interesting conversations. So we'll start out, maybe Paul, by having you introduce yourself. Tell us a little bit about your background before phage and how you got into phage.
Sure. I am 30, almost 35 years in biopharma at this point. I began my career as a management consultant with Ernst& Young and pretty much immediately got assigned to Genentech and have been unable to escape the magnetic pull of biopharma since. I spent probably 10 years doing consulting out on the West Coast before I eventually joined the executive team at Valeant Pharmaceuticals. I was the CIO for that group for some years and as well had a short stint as global head of manufacturing and supply. Um, Left there to join the executive team for Patheon Pharmaceuticals in North Carolina, was the chief technology officer for them, but also ran worldwide operations for all of their what they called their PDS division or their pharmaceutical development services. So essentially all clinical trial material manufacturing and support. And it was at the end of that run. We had a really nice couple of exits there. And it was the end of that one that I decided I'd like to start a company. I had been an executive in residence, helping to support the NC State Poole College of Management, and was able to actually pick up a little thing called CRISPR-Cas3 out of one of the students' inventions that had been brought into an entrepreneurship class. So that was maybe the better part of 10 years ago, and have been the CEO for Locus Biosciences since forming the company in 2015. So I guess you could say I'm a management consultant with sort of an IT background that ventured into both manufacturing and drug development for pharma before taking a swing at a startup. So it's been an interesting ride.
Awesome.
And
Yeah.
maybe you can tell us a little bit. You mentioned that you got involved with CRISPR assets, but what made you think that it was a good time to get into the phage space?
Well, actually, it's interesting. So our origin story for the creation of Locus is actually more of a CRISPR story than a phage story. When we started the company, we actually started it as a CRISPR biotech,
Hmm.
and we had thought that we were first to file on CRISPR-Cas3, um, and we knew we had a payload. What we didn't have was a delivery system. And so we actually started the company with three different pieces of technology from one was from Duke, one was from UNC, one was from NC State, but it was cell penetrating peptides, some nanotechnology and phage. And so the idea of the first year of the company was to really figure out which vector was going to work for the CRISPR-Cas3 system, really. And within like 9 months to 12 months, we figured out phage was probably the preferable vector. And within 2 years of starting the company, we had a data set that was being compiled around our lead E. coli asset that was actually it showed the greatest versatility and started to pick up support from the FDA for some favorable drug development pathway opportunities. And so by the time we were probably year 2 or 3 into the company, we were pretty much 100% phage. So yeah, our story to get to phage was a little bit around some outside buoys. As it turns out, CRISPR-Cas3 is huge. It's just a large complex of enzymatic parts and pieces, if you will. And penetrating peptides couldn't really hold it all.
Hmm.
So they got eliminated pretty quick. The nanotech actually went pretty far, but nanotechnology, it does not have the same benefit as phage in that it actually can essentially get into human cells. And so phage had this wonderful attribute for antibacterial therapeutics anyway, of not being able to infect human cells, and so therefore created this great safety balance. And so we just by virtue of how much you could insert into a phage, um, as well as what we anticipated early days is pretty favorable regulatory support. We jumped in head first I guess after our Series A funding round came through. So that was about 2.5 to 3 years after we started the company.
That's super interesting. I didn't know it was a phage delivery story. That's what we're diving into in the Boyke lab right now is phage as delivery vectors and kind of trying to, you know, actually look at even for human cells now, which would be the dark side. But yeah, like how big is Cas3 roughly? And do you feel like you'll in the future be going down more phages to deliver other stuff or stick with the payload? Like, have you found that the payload is kind of like the more important aspect of the drug or the delivery
Um,
vector?
so we're well beyond CRISPR-Cas3 now.
Okay, awesome.
We just had an IND approved last month for, or I guess it was this month, for Crohn's disease. And that is actually a set of payloads that actually can work exogenously, which is what we were
Ah,
looking for gut delivery to see if we couldn't target and basically eat through whatever is holding this adherent invasive E. coli in place in the intestinal tract. And so
wow.
we actually, I would say, to answer your first question, Cas3, along with Cascade, which is needed to be able to fully express the enzyme, is probably about 7,500 base pairs
Got it.
large.
Okay.
It probably can range up even further for some systems probably get up into the 9, even 9,500. And I think the smallest we ever messed around with was maybe 6,300 base pairs.
Okay.
So large, very large for on and multifactorial, by the way. So it's not like it's one little peptide. It's a bunch of component parts and pieces that need to be reassembled right in presumably the exact right
Yeah.
shall we say, stoichiometry. And then you get this, wonderful added killing effect. And so we certainly didn't start with low-hanging fruit when we were
Mm.
starting to figure out engineered phage. And I think we're exclusively an engineered phage company. We believe that engineering payloads into the phage vectors is the key to breaking out the sort of future of medicine in this modality. And but we don't in any way, at least we do our best to not impact the lytic killing capability of the phage
Mm.
cocktails that we deploy. So we really try to harness and lever a duplicity of MOAs to be able to get to comparability and killing against antibiotics. And that I think is well to be proven out in humans. But I think today we dosed patient 188 out of 288, so we're pretty close to flipping what should be the largest data set of its kind in the phage field. We'll see what the data reads out at.
Yeah. Wow. Joe, do you have something? Because I got millions of questions.
Yeah, just our usual. once we get you to how you started with Phage, sort of, what have you learned? I mean, you've learned a lot, I know, but what are the big things you've learned since the time you started using Phage to now?
So, what I think is maybe a little interesting about our origin story, and you know this background, Joe, and maybe most people in the field do too. But so, Jessica, we came out of NC State, um, really hot and heavy on CRISPR-Cas3 systems, and then um, pretty quickly engaged in fairly large strategic partnership discussions. Johnson and Johnson was the primary anchor, but there were some others. And it wasn't until we acquired EpiBiome, who had been working really for years on high throughput screening, liquid handlers, as well as machine learning for various types of engineering to phage. It wasn't until we put the two companies together that we actually broke out and got that Janssen deal, which I think was sort of industry setting at the time, $800 million deal. By the time the whole thing came crumbling down, we were able to get about $85 million of
Okay.
funding from Johnson and Johnson on really four key indications across both Pseudomonas and staph. So it's been fantastic being able to sort of leapfrog off of that. But I would say, Joe, that the answer to the question lies in that technology move. So we, I think, like most, had assumed that you'd discover great phage down at your local sewer treatment plant, fish them out,
Hmm.
basically characterize them, stick them in a cocktail, and go. What I think the guys at EpiBiome did, Aaron Hammock and Nick Conley, pretty ahead of their time, was to basically move to an automated robotic platform to discover phage in mass, characterize in mass, and then use essentially early stage AI to figure out how to put those things together, looking at things like burst size, receptors, I mean, you name it, and trying to figure out how do you put together a cocktail of phage that's comparable or superior to an antibiotic. And that to us is that's really what we believe. We believe that discover thousands of target phage against a target species or genus, characterize those in mass, figure out which ones are ripe for engineering, which could be ones others easily eliminate, right? Ones that maybe don't have the greatest host ranges, but have some property we really want,
Hmm.
like a receptor overlap or something along those lines. Engineer a payload into it to get its killing efficacy above its natural wild type counterpart and put it together into a fixed cocktail. And if we're right about it, you end up with a drug that ends up in a single vial, could be lyophilized down into a powder and brought to a pill so that you can eventually get to a pill to pill replacement for antibiotics, which would arguably change the world, right? From broad spectrum pathogenic treatment to precision medicine, which is which is exciting, I
Yeah.
think.
Yeah. It's always been fascinating to see. I feel like Locus has done a lot of things. I think a lot of people say this about Locus, that you're doing things in a very practical way and like also things that you wouldn't think that people would innovate on. Like I remember the phase 0 trial that you guys were talking about at conferences in years back, probably Nick talking about that, about how you went out and figured out like in advance of the trial itself, like which of are the patients? Can we actually get enough patients with this profile that like our drug hits? And I'm probably not fully understanding like the ins and outs of it, but I just remember like I always remember that like wait, we always think about phase 1, 2, 3, And why not before one? And like, so has that panned out well and like led to the fact that you're able to kind of move towards successful trials, do you think? I've always wanted to ask that.
Yeah, well, I think we're firm believers in phase zeros. It's expensive. Usually people can't afford them, but you essentially are trying to both validate the target, hunt for a biomarker that you can use for enrollment, but also vet your facilities and make sure that your target patient population exists before you go into the larger, more expensive trial. You do not need FDA approval to do a phase zero. You do
Interesting.
need FDA approval to do a phase one. So I would say 60 to maybe even upwards of 80% of the sites that we used in the phase zero pulled through to the phase one. And the translation of host range against our cocktail. You guys know all these words. If I lose you anywhere, feel free to stop me. But
All good.
so we were looking at, I think, 94% host range coverage or sensitivity to the cocktail. That translated across was about a 200 patient phase zero data set. So far, the data that we can report in the unblinded PKPD uplift portion of the trial, alongside actually the phase 1B that we also did, they were also both 94%
Wow.
sensitivity. So it had translated from robots to phase zero to phase one to
Yeah.
PKPD uplift, and then we'll see when we unblind the phase two what the numbers look like. But we assume we will get that same host range, and that's part of it. So are you? I think maybe one thing we think about is, are all of the phage that you're going to find for a particular bacteria around the world the same? Like, are those actually the same? Are those maybe different by geographic region? Are there bacterial strain differences that would enable the phage to evolve differently? And I think what we're beginning to lean into is yes, that it is likely there are some geographic differences. And so, is the United States even large enough that you might see differences from coast to coast? We don't 100% know the answer to that, but we suspect yes. And so maybe tying a few things back together, to be able to hunt for 10,000 phage, characterize them, put them together in a cloud, figure out which ones go best together to get to 4, 5, or 6 of them that go into a fixed cocktail, to us is critical to being able to make sure you can translate, and then getting out there in the field and then going to physical locations and getting some potential patient samples, spraying those down with drug product, making sure you have translatability. I mean, that's just good trialist homework to make sure you can be successful. There's nothing more deadly to a biotech company than planning to have a trial take 2 years and it take 5. And especially in the phage arena, it's such a low cost, a low, like, literal financial, like, floor to be able to get into the industry. I mean, I think a lot of people have had massive difficulties once they hit the clinic. It's
Interesting.
expensive. It's very hard.
What do you mean by a low financial floor for our phage research scientists audience, but who are
Yeah,
curious about the business side?
I mean, I think, this, Joe and I have talked about this in the past, but, I guess, I'm not a huge fan of the Cystic Fibrosis Foundation approach of sort of $3 to $7 million to go conduct a 5 to 10 patient single facility or dual facility trial. And I think that Pseudomonas data that's come in the last, shall we say, 4 or 5 years has been a massive momentum killer for our space.
Hmm.
So in other industries or sectors, I would say, so let's say you're working in immunology, you're not you're not getting into the clinic for $3 million. You're not sniffing the clinic for $3 million. The sort of normal path to get into the clinic um, is I think just more advantageous in phage because of the safety profile. And so you don't need as much capital raised to get a theoretical drug into a trial. And that, um, having been in the space for over 30 years, this is where most of us fail,
Hmm.
is the clinic. It's expensive, it's difficult. You may have the best drug in the world, but if you didn't set up your protocol right, and you don't understand your target patients, and you don't have the right KOLs, and you don't have the right sites, you may never enroll. Or if you do enroll, you enroll the wrong patients and you get bad data and you don't get a second chance.
Hmm.
So that's, to me, I think a huge, huge headwind in the industry is the one that everybody thinks is a tailwind, which is the low financial barrier to get yourself from an academic lab into a clinical trial setting. And I think I don't know of any data that's helped the industry break out. And
Interesting.
that is one of the largest challenges in our space. Because why can't we attract $400 million
Mm-hmm.
rounds?
Mm-hmm.
That's a good question, right? Because other people are attracting $400 million rounds. So why aren't we attracting $400 million rounds? With our safety profile, how come we're not attracting $400 million rounds? I think it's because there's been some damage done by essentially the creation of data sets um, that aren't great. They show promise, not for a private equity firm or for a major venture capitalist, but they show promise. Um, and that I think is something we really, really hope at Locus that we blaze a new path and that it opens up funding for a lot more than just us. There's way too many targets for any one company to be able to absorb. So, I think that I think is, well, an opportunity and a challenge.
Yeah. Oh, that's such a
So
fascinating
one of
perspective.
the things you didn't talk about in that was endpoints. And one
Mhm.
of the things I feel that you guys have done really well, and I've been lucky enough to hear some of these discussions, is how you go about figuring out the exact endpoints. Because I think that's another point where people can really screw up their clinical trials is by having unrealistic endpoints or anyway, you want to talk about how you guys go about thinking about endpoints?
Yeah, I mean, I think we start with our secret weapon, which is Dr. Kim. Paul Kim is 30 years in industry developing drugs, background in places like Eli Lilly and Amgen and others. Paul is, I think, quite familiar with how to best manage endpoints to the phase. And so the earlier the phage, the obviously the like least amount of rigor from an efficacy standpoint with maximum amount of rigor towards safety and tolerability is, I think, the way that people would normally go if they're trialists, leaving the difficult, maybe microbiological endpoints to exploratory instead of primary or secondary for at least as long as you can. Um, I think we also we I think have been quite unique in trying our best to not guess at a dose, trying to figure out duration and volume of the dosing regimen as it's tied to your titer is really important to get your exposure right. And this is very difficult, but I mean maybe this goes back to some of the other trialist comments, but if you can get simple PK plus tolerability, and those can be your primary and secondary endpoints, and you put microbiological cure as an exploratory for a phase 1B type of structure, you should. It'll be maybe harder to go get $100 million after, depending on how you design the trial or what type of control arm you may or may not have or could afford. But I think you're trying to make sure you get the drug where it needs to be. And I think if you're relying on the lytic cycle, you also want to make sure that your initial rounds of infectivity are absorbing, which would be measured in later time point amplification insights. And so, PKPD is how drugs work. And I think paying very, very special attention to PKPD in early stages till you get it right, you really shouldn't progress. Because your chances of failure are pretty high. You could have the right drug in the wrong quantity at the wrong time points, and the right drug would fail. You could have a mediocre drug where you perfected the exposure and it might work. So which is best? The latter's best because the latter progresses, the former stops. So it's like your endpoints are tough. When it's not a one-person game, right? It's somewhat of a negotiation with the FDA. You have to follow the guidelines, and you have to be able to gain their alignment on the protocol, and not just your endpoints, but your enrollment criteria and how all those things mix and match.
Yeah. No, this is super interesting. I feel like we should click in a little bit more to this kind of like the PK side, and also when you guys have the approach of having thousands of phages and then down selecting from there. And I wonder, have you found that there's innate properties of phages themselves that make them better likely to have a good PK profile, like they just handle the serum better or whatever? Or is it, do you have more of a lever in the formulation of the phage and things that are like not phage specific?
I mean, I don't think we know anything that's secret in that arena. I mean, maybe we do. We get pretty high titers compared to most. I think it's not it's not so much an individual phage, in our opinion, as the overlap of phage. And in fairness, we suspect. We don't know. We won't know till we flip this next set of data where we actually have a large placebo cohort. And maybe, okay, we think it's really important to get as close to acute infections as you can, to stay as far away from chronic as you can, as you're trying to prove something out, like PKPD. Chronic bacterial infections, I mean, take CF in lung. I mean, that's a polymicrobial, soup of very colonized and multi-pathogenic strain/species environment. I couldn't be more difficult to get down into the nooks and crannies of the lower respiratory tract. And so which phage do you choose? Maybe the ones that actually have better anti-biofilm characteristics versus, let's say, depth of kill. So your target environment, the organ you're going into, how you're going to get it there. Like those are the things, Jessica, that I think when you're picking a phage, you want to actually look at the grouping of what you have to eat through to get to the pathogen. And that's undoubtedly one of the hardest parts. See, we don't need phage therapy for regular routine infections, right? Because we have these less than a penny per pill antibiotics, which is problematic. We're only really using phage to go after the nastiest stuff we can find. So it's a really difficult target base. If you don't go after that really nasty stuff, that's obviously a much different regulatory pathway, a much larger patient count, likely superiority designs, those types of things just drive up the costs, which we in this sector have been yet to be able to fundraise. Anyway, I'm meandering over like 10 different subjects at
Yeah,
once, but I
that's
think
great.
which phage go together has a lot to do with the environment in the body that you're going up against.
Yeah.
Yeah, I
Okay.
mean, I guess chronic infections almost by definition have a degree to which they're persisting, and obviously persisters are tough to deal with for a variety of reasons.
Yeah.
So
I think
I'm
that
gonna go ahead.
I think that to me that's the holy grail is intracellular pathogens. I think
Oh
if
yeah,
Intracellular.
I think if we can use phage therapy to address precision medicine needs for intracellular pathogens, the modality is here to stay. If we
Hmm.
can do that, and because that is specifically an area where antibiotics do not work, and likely one of the greatest problem areas of multi-drug resistance infections. Um, but I think that can we suspect that can be handled through payloads. Um, it may be different regulatory pathways, but I think we think it can be handled through payloads. So anyway, sorry, Joe, I didn't mean to stop you from
no,
your pivot.
no. I mean, I think I don't know what else I was going to say. Jessica, do you have more
Yeah.
questions?
Oh, yeah. Well, I'm just kind of thinking about. I think people a lot of times think biofilm, and they don't think intracellular. Like, I mean, so we're talking about like there's biofilm. I think we're in the urinary tract infection context with what you guys are targeting right
So,
now,
we,
right?
so I'll give you some interesting data that we've been using it. So I think something like 65% of the UTIs in the phase 1B and PKPD uplifts portion of our UTI trial they tested positive for adherence and invasive characteristics.
Okay.
So it's likely, would those be under some kind of protected biofilm area? Probably. But are they intracellular pathogens that are inside the bladder wall? Is that where these recurrent infections are coming from? I mean, we've been going to ESCMID for a number of years, and I think the general shift in thinking has changed a bit, at least in the years that we've been going to the conference. In the early days, the idea was that most UTIs might be being caused by reinfection,
Hmm.
MDR hangout spots in the intestinal tract that somehow through either a fissure or poor hygiene make their way into the urinary tract. I think we've been on the other side of that argument in that we think it's likely colonized bacteria that is potentially intracellular bacteria, either from a
Wow.
previous infection event that didn't quite go away in full, or potentially, just an injury that was a past injury that you just haven't been able to
Hmm.
to fully clear. That you get a UTI, you get treated with antibiotics, it beats it back a little bit to the point where you feel better, but doesn't get rid of it. And at some point, it begins to shed, bloom, and you got a reinfection of it.
Are you? Are there intracellular biofilms? I've never contemplated this.
Um, I don't know if I would say, well, I'm probably not the right person to ask that, but I don't think we think about it that way.
Okay.
I think we think about biofilm areas on top of
Yeah.
intracellular pathogens, which are on top of cells that are dying, that are shedding their bacterial loads, and new bacteria that's somehow in there. We typically think of like a soup.
Yeah.
Um, where it's all in there. And what you want to be able to do is get your exposure right, so you're not just clearing what I kind of call it transactional bacteria. So it's the stuff that's likely causing a problem is the non-transient bacteria. That's the stuff that's probably causing a problem, the stuff that doesn't go away. And that there's I guess we know a lot about this from all of the Crohn's work we've been working on for years. So our theory of one of the causal factors of Crohn's is that intracellular pathogens are what your body's immune system is actually attacking, and they are surrounding the lesion areas that Crohn's patients get in their intestinal tracts, and they just get worse and worse, right? Everybody's got E. coli in their guts. Not sure why some patients have that build up become adherent. Not exactly sure how adherence leads to invasion, but your end result is probably transient, adhesive, and adherent invasive E. coli that's in your wound areas. And if you massively debulk that bacteria, do you get a corresponding decrease in the immune system's attack? And that theoretically is curative, not suppressive, which would be, that's why I go back to, I think intracellular pathogens are the holy grail for the field.
Yeah.
If phage can clear intracellular pathogens where antibiotics cannot, we have a new field of medicine.
Fascinating. Yeah.
And dealing with pathogens is maybe the tip of the iceberg, because inflammation in the body, in theory, is the cause behind a substantial amount of chronic disease, for which we in society use suppressive technologies. So, immune suppressants for everything from psoriasis to Crohn's and colitis, right? And you're not actually curing anything. You're just telling your body not to attack yourself. And then I mean, you're probably preaching to the choir, but I think this is one of the like dirty secrets of our industry is we're not really hunting for many cures, although it's changing. Certainly, gene therapy's been making huge strides. Definitely got to get our delivery vectors right on that front. But going after genetic disease, great. But I think without question, I think bacteria in the body is certainly causing inflammation, joints, I mean, intestinal tract, and in theory, if we can precisely get the bacteria out that's driving the inflammation, we're going to see a lot healthier society.
This is a really great angle because a lot of our audience, I feel, is on the phage research side, and I think they want to know like what's the most important research that they could actually do that would move the needle. And our podcast, we really want to talk about like what's stopping phage from being in the clinic, and so I'm glad that we kind of are landing on this like one kind of concept of our phages kill great in liquid culture, whatever, LB broth, but they actually need to get not only through biofilm, but into eukaryotic cells and do their killing there. Like, I don't think that that's talked about much in the phage field as
Yeah, and
like the holy grail.
yeah, I don't think a wild type phage biologically can get into an intracellular pet. I mean, Joe, you probably know this better than I do. What we're assuming is that the payloads can get to either a happier and healthier macrophage environment, so that macrophage that should be doing their jobs actually do their jobs. Um, and or straight delivery of maybe some peptides that do the job for you, and get to those pathogens behind the cellular wall. But that, to me, that's the power of not just the payloads, but the phage cocktails. And that, we didn't really touch much on that, but I think individual phage that eventually hit, a more rapid resistance profile than what you would want for anything other than personalized medicine. I think we have to try to, if we want a large scale investment into the space, I think we have to be thinking about large scale commercial drugs to attract that investment. And that it's hard to see how personalized phage therapy, I mean, it's super important to advance. So I don't want to say the wrong thing, but I don't think we're attracting um, large scale corporate strategic investment through that form of personalized medicine. And so cocktails are important, and the payloads that go into those cocktails are equally, if not more important. And I think that, I'll give you a great example of an idea we had from something that was rejected, not that long ago, but it was a really good idea. Can, do cancerous tumors have a specific microbiome profile? And can you find phage that can amp off that microbiome profile to deliver a toxic payload that would kill that tumor in place? Could you do that? Could you use phage to do? I don't think you're getting the easy clinical development pathway that the FDA is saying is available to phage therapy today for that. But could you do it? Could you deliver a drug that only amped its payload at the place you need it in the body to eat away a tumor? We think you can. It's tricky.
I've heard about this field of work a couple years back, and I wondered, did the whole tumor microbiome field debunk that, or is that still a thing that there is a tumor microbiome? That part I'm hazy on.
So I think microbiome was a field that actually got that massive amount of investment, and then the initial waves of clinical results failed out and or were mediocre, and it cooled the whole space. But I mean, I think it's important, especially for people that are in the phage research field, to understand the dynamics of that microbiome environment surge. So the really the first generation broad spectrum sort of fecal transplant oriented additive microbe approaches got billions of dollars, just flat out billions of dollars. And when you really think about, like, okay, you're going to add in copious amounts of a bacteria and hope that it adheres, that invades. Why would you add? Because it's easy. That's why you add. Why do you go after cystic fibrosis lung infections if you're in the phage space? Because the CF Foundation gave you $5 million. So you have, like, what, but the microbiome space, I think, is just basically collapsed under its own early stage quick win philosophies, I guess is the easiest way to say it. And the thing that I think is important in that is that I think the microbiome is quite real. And I think subtractive prior to additive joint therapies are likely the future of medicine. Carve out a niche.
Yeah.
Get out what you want out, and then put something back in there that you want to adhere to maintain. Don't just drink the maintain every day, assuming that the bad is going to somehow be out competed I don't know, no one knows, but I don't think that that theory of add only works. But
But I guess it's worked fairly well for C. diff.
so C, well, we all have C. diff, right?
Right.
So C. diff in its base form is there, which might not be that far, Joe, from other things that we want to have come out. But I think in that particular case of basically adding in a bunch that essentially competes with the C. diff bloom, you're doing that concomitantly with massive amounts of vancomycin. So you had to subtract
Hmm.
that was in there to make. In fact, all microbiome therapies typically have a front runner of antibiotics. Most gene therapies have front runners of antibiotics that blow out your microbiome right before you dose somebody with a multi-$100 drug, which is interesting in and of itself. Um, but anyway, we digress a little bit. I just think it's important for people in the phage space to not give up hope on the microbiome.
Yeah, that's super
I
interesting.
think that I think that the microbiome is very, very important. And the idea when I first started 10 years ago, the idea of trying to understand the microbiome, just the fact that there were trillions of cells in the human body and different by each person, just the math of that was very intimidating to people. Do you think we should be as intimidated now with AI? Is anybody that intimidated now?
Hmm.
The money's gone.
Hmm.
That's the interesting thing. Was it timed right? Because the microbiome is seemingly like a ripe AI problem
Yeah,
from
totally.
my perspective, which is
Yeah.
another really cool way to look at because there's the phagome goes with the microbiome. Are they not the same thing?
Hmm.
You know, I'm sure there's other people who've done this, but there's a friend of mine who's at the University of Oregon, Karen Gilliman, and she's done, I mean, she works on zebrafish, so you can do a lot of things with the microbiome. And she has a modeling collaborator, and they, at least when I talked to them 5 or 10 years ago, they were doing all this stuff where they were introducing into notobiotic fish like specific microbiomes and like predicting what was going to happen and then seeing what the answer was.
Yeah.
And that, like some of that data, going back and looking at it and mining it with AI. I mean, obviously, fish aren't exactly people, but they do have a very similar, immune system and a lot of similarities.
Yeah. Well, it's also difficult. The microbiome is also really difficult because I think the most is studied from basically stool samples or whatever you excrete, right?
Right.
And I think that that's at best what might be shedding
Mm.
or is transient, which is unlikely to be the problem. Well, the
Mm-hmm.
shedding is if you can get it in a, but since we kind of do, well, in this particular sample, what are your greatest masses of different types of bacteria to determine what's in there, not sort of what's everything that's in there. You
Mm-hmm.
really have to get in there and do some biopsies and understand the biopsy outcome from certain problem areas. Like think of lesions for a Crohn's patient. Like, can you get actual tissue that's close to, which is its own trialistic problems. But that I think is where we're going. I think understanding tissue and the surrounding microbiome and phagome environment at that point of problem, like that I think is a math challenge we can handle now.
That's really
And
cool.
so, like with time and data, I think you'll have a whole new class of drugs that spawn up out of that. And I just encourage people in the phage space to stay after it because we're likely the ones that'll be there first. We may not stay, but we'll likely be there first.
Yeah.
So this has been great. And we're getting close to the end of the hour, so we should probably give you our sort of final wrap-up questions. And you've probably touched on some of these things, but what of these things you've talked about a lot of future-looking things, but what's sort of realistic in 5 years and 15 years, and what do you think are the biggest barriers to keeping Locus and other people from getting there?
Yeah, well, what we're finding on the Crohn side for sure is patience.
Hmm.
The ability to get sites and KOLs to PIs to sign up for trials in light of the just massive amounts of billions of dollars going into research in that space, like finding patients for things that are non-pathogenic, that's going to be very difficult. And I think it's going to need to be more in that 15-year bucket, Joe, than that five. And that it's just going to take time to be able to break that open and get the PI attention. Um, I think infectious disease and pathogenic threats and really understanding where antibiotics are not going to work and how to push what we think are engineered phage cocktails into those gaps, that's the next 5 years for us. And so we've got, our lead barter program for UTIs, um, fairly hot off the press, but we now have a Pseudomonas asset advancing with the NIH and the NIAID division for hospital-acquired pneumonia. And we have a blepharitis indication for staph advancing with Viatris. And so 3 different body parts, all MDR infection sites, 3 different sets of payloads, and 3 different routes of administration. So you're trying to figure out in the next 5 years, how do you get the right exposure of an engineered drug product to the site of infection to clear it? And how do you work concomitantly with antibiotics to make sure those infections never come back? That, I think, that opens the field. So the next 5 years is proving that commercial grade products will work and hopefully drive a profitable company. The next 15 years, I think, is anti-inflammatory chronic treatments that span across into, human genes. And we get into things that probably nobody's thinking about right now.
Yeah, I
That's
think
what I
that
think the future's got.
awesome. I think that tie to the inflammation field is, yeah, such an interesting one to unlock and like blast it all the way open. Because I feel like when phage gets lumped in as like antibiotic replacement, it's like a tiny market and everybody wants to, at least I was under the impression that that's kind of why phage companies haven't raised much money, is because that's not an attractive enough, big enough market.
I think I think it's actually infectious disease and the MDR, if you label the things correctly, I think it's a pretty big market.
Okay.
I don't know if the pricing of those drugs, I don't think that the economics of it can handle the prices that have been coming.
Yeah.
And I don't mean for phage, I mean for other MDR treatments. And so even on the small molecule side, certainly the large molecule side, you start getting into 25, 30, $35,000 for a particular treatment. You're going to get the keep behind glass break when finally necessary. If you want to get into dosing millions of patients, you got to get a price point somewhere that enables the hospitals to put that drug into a non-break glass when needed scenario. And I actually think going to the phage side of things, I think phage has the COGS potential to get a priced product that could be concomitant therapy that could break back open millions of patients. And that the financial dynamics on that for a drug that's priced anywhere from 1,500 to 2,500 bucks or something, maybe the cost of an iPhone, that kind of thing, I think is possible with phage. So it's unlike any other large molecule in its production capacity. It's It's quite contagious, as most researchers would know. And so making it is it's interesting, but definitely doesn't have the same cost dynamics as other large molecules. So it's an advantage if you can figure out how to harness it.
Yeah. Okay. Well, that's very encouraging. And so your investors must feel this way at least some degree. And it's not all doom and gloom on that side, that antibiotics is a broken market and there can never be a new one.
We don't think that at all, actually. I think there's a timing element of this. I mean, I do think antibiotic resistance is growing, and in some areas exponentially. That'll continue to grow. And I think that's, the it's possible to get in front of it financially. It's just a matter of actually kind of pricing things properly to break it open. So we'll see. We'll see
Yeah.
where it goes.
Yeah. Thank you so
So
much.
I just came up with one last thought, and I know Jessica's ready to wrap us up. But
All
yeah,
good.
have you guys thought about VAP and like giving people who are on ventilators for a long time phages prophylactically? Do you think? I mean, I think that might be a really hard business market, but if you can encourage, I mean, hospitals might. I don't know.
So we are thinking about VAP because with the HAP indication, I think 20 to 25% of HAP patients advance to VAP. And we are thinking about the physical device components of the ventilator and the tubes and what that means and how we would get in front of it. We have thought about prophylactic products, Joe, and I think they're definitely on that price spectrum. The lower you can get the price, the more you can probably get the pickup. Um, in that, I think there's certainly some. I'll tell you an area where we think about it all the time is colorectal cancer, because the sort of environment of E. coli and Bacteroides and all kinds of other crap that's in there that is a long range exposure issue that people believe long range exposure to those bacteria create the environment that's ripe for colorectal cancer to evolve. But long range prophylactic, especially if you're genetically predisposed to colorectal cancer, if the price of that was correct, it probably could pull it off. It's just a very difficult trial. It's a very long, very expensive trial to get there, but then you'd have a dominant market position. So I think the way we look at the commercial aspects of this are, that's right. I'm sure you've heard this one, but I mean, people have always looked at us with like four eyeballs, like, why are you going after UTIs? Why didn't you go after the purpose of UTIs is just millions of patients of recurrent UTIs, and the suspected cause, or one of the suspected causes, is MDR. So we have this beautiful balance between commercial opportunity and regulatory help to advance and get there with some reasonable amount of cost. And so, we look at all the things we've built from a platform perspective and think about, well, how many drugs can come off this platform? First one, it matters. It matters a lot, but what matters is the data. It's not necessarily the full commercial, opportunity by any stretch, honestly. And then you can look at infectious disease as a stepping stone into things like immunology, and immunology is a stepping stone into oncology, and oncology is a stepping stone into neurology. And how many of these different areas could have precision microbiome that is eventually part of our mainstream medical treatment? I mean, do you think 100 years from now, somebody's going to be balancing your microbiome to optimize your longevity? Because that's what you're talking about. I'd say yes. So we're between now and 100 years from now, we'll all be dead. But from here to there, how do we break through?
Yeah.
We blew way past our 5 and 15 years.
Got 100. We got to bring that in.
All right.
Awesome.
Well, thanks a
you.
lot, Paul. This has been great.
Sure.
I don't know if Jessica has another question or not.
No, I'm good to close this out. Thank you so much. What do you need help with from the phage world, or what would you like to direct people to that you're excited about, or anything that we can leave them with?
I mean, we're always I think we're always looking for talent. Um, and we're primarily our labs and manufacturing are based in North Carolina, so that's
Yep.
always interesting. We hear a lot from folks from places that maybe don't want to come to North Carolina, but for those that want to
That's an
come to North
awesome state.
Carolina, love to be able to network, and love to be able to actually collaborate with others as well. For what it's worth, we love
Yep.
payloads. And so
Okay.
if you're
Love
out there and
payloads.
you're thinking about things to work on some payloads. And
Okay.
maybe you can sell your payloads to Locus or anybody else.
Yep.
And then I think just as a general word of encouragement, I mean, I do think the US government specifically has a pretty solid track record of making large investments to try to help people advance platforms that could change things dramatically. And I would just encourage as much government interaction as you can get. We found the FDA to be incredibly partner oriented. I've never seen the FDA so collaborative as they are in this space. It's really cool to see.
Yeah.
It's hard, but it's cool to see. I've never really, until this job, I've never really gone deep in with health and human services in so
Mm.
many areas between the NIH and between BARDA and the NIH has been great. And I think the Department of the Defense has obviously had a well-worn track record investing in this stuff, and that those dollars matter. So if I was at one of the bigger labs, I'd be trying to chase some of those because the more funding scale you can put to your endeavors, the better the assets that come out. So I
Yeah.
think it's a very promising field. I think we've been around for a long time, and I think we have a bevy of mediocre to poor data that now we have to weed through.
Hmm.
But for those that are maybe a little bit more longer range in their vision, I don't think you have a safer delivery vector to get something into the human body that's better than phage.
Hmm.
It can carry a mass that other delivery vectors cannot, and it does not infect human cells. And so, what can you do with a phage?
Yeah.
That's a really exciting question.
Yeah. Cool. Awesome. Great. We'll leave it at that. Thanks so much, Paul.
Yeah, absolutely.